Okay, today we're going to do it a little bit different today. So we're starting module 3. Module 3, I kind of need to use a computer. So I'm going to do my best to teach you guys here on the computer and record via Zoom. Hopefully everything will work out okay. Okay, so today we're going to discuss cloning a gene. When I teach on the computer, I tend to only look at the computer. So if you have a question, just shout it out or get my attention because I might be zoned in on the computer. It's a bad habit. Okay, cloning a gene. We're kind of going to go over the steps, and I'll show you precisely actually how to design this, how to do this. So first, let's understand the strategy. Okay, so the broad strategy here is there's some genome, and on some genome, there's some gene that you're very interested in. Maybe you want to express this protein. Maybe you want to study this gene. Maybe you want to make some mutants of it. Maybe you want to do who knows what. Maybe make a transgenic insert. Who knows? But you're interested in this gene, so you need to recover it and put it on your plasmid. Okay. So the most simple strategy to do this, okay, this is, keep in mind, this is going to be applied to prokaryotic genes. So this exact precise method wouldn't necessarily work on eukaryotic genes because they have introns and exons. Okay, so we're talking about simplest way to clone a gene in a prokaryotic gene. Okay, so the simplest way to do it is you design primers. Okay, so your primers are going to be essentially right at that start codon, ATG, and right at that stop codon in that gene. Okay, so they're PCR primers. When you design these PCR primers, you're going to add in restriction sites into those PCR primers. So basically, you're going to have one primer. Here's your forward primer. Here's your reverse primer. The blue part is the part that anneals to the gene. Okay. Get a different color. The orange part, what are you going to put on that orange part? What do you need to be able to, so the PCR is going to amplify this genetic product. Okay. And then you need to be able to insert it into a plasmid. So what do you need to add to the primers to be able to do that? So I've talked a lot about cutting genes out, pasting them. What were you going to say? Yeah, you got to add your restriction enzyme sites. Okay, so you're going to have to add restriction enzyme sites. So let's call this 1. Let's call this 2. And that's not it. You also actually have to add a little bit more. So here, the green section. Green section is essentially 4 random bases. So it's 4 extra bases. And you need to add this into your primers because eventually when this product gets made, so let's say this was your amplicon, when you add those restriction enzymes, let's do restriction enzymes the color of purple. Okay, so here's a restriction enzyme that's going to grab onto the DNA and cut. When you add the restriction enzymes to actually cut this, they need to be able to grab something on the 5' end of that restriction site. So there needs to be something right around in that region. Otherwise, it won't be able to grab. Okay. So the reason you have to add that 4 random base pairs is because the restriction enzyme is going to grab onto like this, and then it's going to make that cut. And if you don't add those, if there's nothing there, it won't be able to cut efficiently, and you'll get real sloppy cloning. It won't really work well. Okay. So essentially your primers are going to look something like this. You're going to have a section of your primer that binds to the actual gene. You're going to have a section of the primer that is building in the restriction enzyme sites into your amplicon. And then you're going to have 4 random base pairs on both the forward and the reverse primers. Okay, are there any questions on sort of most simple primer design? Okay. So that's in a nutshell what we're going to do today. Okay. So for step 1, to design these PCR primers, what do we need to design the primers? Let's actually do it. What do we need? A sequence of what? Which is what? the gene X. Okay, so what gene do you want to clone? And where would you get that sequence? Where can you get sequences of genes? Yes. Okay, so one, we need a sequence of gene X. Okay, I've done this, or we're just gonna pick a gene. Let's just pick REC-A because I know I'll be able to find it. So REC-A is a recombinase from E. coli. So this is gonna be the name of our gene X, REC-A. So you're gonna go to the internet, okay? NCBI is the National Center for Biotechnology Information. So all the genomes that get sequenced with federal funding get put on this website. So many of the gene sequences can be just directly pulled from this. So if you want to navigate this website, which you should all learn how to do, it's an extremely important website. What we want to search for is a drop-down tab, a bunch of things you can search for. Let's search for a gene, and let's say E. coli REC-A. So the name of the gene is REC-A. Let's see if we can find it. Okay, we found it. So here is REC-A. This is the gene from Escherichia coli strain K12. That's a common research lab strain. So let's click on this. Okay. So a couple things about NCBI if you have not ever looked at it yet. So this viewer browser is very useful, and you should start kind of browsing on this website and learning what you can do and what you can look at on this website. So this picture right here, this is a picture of the chromosome, and this is the genetic context where REC-A is sitting. So if I zoom in on this, so you can see here's the chromosome. It's just a subset snapshot of the chromosome. And you can see that REC-A is surrounded by some other genes. And when we talked about the prokaryotes in the prokaryotes lecture, we talked a lot about how their genes are very crunched together. Oftentimes they're in the same direction. So you're kind of seeing that here with this pattern of genes on the chromosome. Okay. So if you look down further, scroll down, here is REC-A, the actual gene in this little, I don't know what you call this, genetic browser thing. Okay. If you keep zooming in on this REC-A, you can keep zooming in and eventually it will give me the actual sequence. Okay. So you can actually zoom in far enough where you see the actual sequence. Let's see if we can find the start codon. Will you open that door? So here I'm just zooming in. I'm trying to actually find that start codon. There it is, ATG. So you can zoom in so far where you actually see the sequence, and you can zoom out farther and farther and farther to see the whole context of the chromosome. Okay, so this is the gene we're interested in, so we got to pull its sequence. So if you mouse hover over the gene, you should be able to get some options. One of these is called FASTA record. Okay, let's click on that and let's see what happens. So this is the protein. That's not actually what I want. Let's try. Let's try GenBank record. Let's try this. Problems. Let's see what happens if I click this. Hang on. It's still giving me the protein. Watch me now fail right in the middle of my tutorial. Let's try download FASTA. Let's try this. Download FASTA. It's still giving me the protein. This is very frustrating. You have to be able to click the right buttons to get the gene. Let's see what happens if I just hit FASTA. Here we go. Okay, so all I had to do was hit this button here, FASTA. So that is actually one common thing that's very annoying with this website is there's like a billion different buttons. And oftentimes you have to just tinker around and click around until you finally get the right button that you want. But we're trying to get the actual sequence. So what I did is I clicked FASTA and it pulled me to the sequence. So here's the gene sequence for REC-A. How do you know this is a gene? Look at the sequence. How do you know it's a gene? What definitive features of this sequence tell you that it's a gene? Look at the letters. Yeah. So you can tell immediately this is a gene that it worked because here's ATG, here's the stop codon, TAA. Okay, so that worked. Other things about this is a very common format where you have this little caret and then a bunch of like information and names and then carriage return, which is a return to the next line. This defines an actual computer format. This is called FASTA format. So FASTA format is the formatting, the syntax for how you describe strings of genes, either in protein or DNA sequence. So this is the FASTA format, okay? So I'm gonna take the FASTA format. I'm gonna pull it up here in this little text editor. I'm gonna paste it. And what I'm gonna do is I'm gonna quickly just remove all these carriage returns with nothing. So I've removed all the spaces. And I'm just gonna pop this into a Word document. Okay, so this is kind of how I'm gonna work within this framework to design our PCR primers. Okay. So we've captured sort of like the genetic sequence. This is what we want to design our primers from. So let's go back to this. We have our sequence of REC-A. What's step 2? What do we do now before we design our primers? Or maybe we do design our primers next. What do you think we do next? Yeah, that's exactly right. So we need to pick a destination plasmid. Okay. So because we've talked a lot about PBAD and we're very familiar with this system now, we're just going to pick up, our plan is going to be to clone this into PBAD. Okay. So I pulled the map of PBAD. Here's the PBAD map. I need to kind of make a decision here before I design any primers. What do I need to decide on? There are some decisions that need to be made about this plasmid. Where is the gene going to go? Is the gene going to go right here? Where's the gene going to go? The multi-cloning site. Okay, now that tells you right away, like we need to make a decision. What decisions do we need to make about the multi-cloning site? What are these? These are restriction enzymes. How do you know which restriction enzyme you want to use? Yes. Okay. So there are some considerations here to consider. One of the things that was said is each of these restriction enzyme sites has a custom cut site. Okay. You don't want to pick a restriction enzyme site that has a sequence inside of gene X, because then your gene X is going to get cut in half. Okay. So that's one thing that we need to check is we need to pick restriction enzyme sites that don't cut the gene X, in this case, REC-A. Okay. The next other question I have is how many restriction enzyme sites should we pick? How many do we need? Not Ben. How many restriction enzymes do we need? We need like 5? He shook his head no. Why? Why'd you shake your head no? That we don't need 5? No, not you. Yeah. You need 1 on both ends. Okay, good. So do you want 1 or 2? Why 1? So let's think about that. So let's draw what's going to happen. So what was said was here's a plasmid. Here's the multi-cloning site. Here's restriction enzyme 1, and here is restriction enzyme 2. In theory, ... there's more. Okay. And here is our amplicon of REC-A. We need to, what was suggested was we could put a 1 here and a 1 here. And then we could cut our amplicon at the 1. We could cut our multi-cloning site in the plasmid at the 1. What will that yield? So when we clone this in then, what will be the product or products? Okay, so you're going to have a plasmid. It's going to have a 1, a 1. You're going to have rec A, a 1, and a 1. Okay. So do you see how this can fit here? This can fit here. So one product is going to be this. What's the other product? This is product A. What's product B? That could also happen. Yes. Okay, you're right. Okay, good. You could also get this because this site matches that site. Okay, that could produce that. There's also a third product then. What's the third product that you could get? Yeah, the gene could be flipped backwards. You'd have rec A like this. Okay, given that the promoter on this plasmid is PBAD and the promoter looks like this, is this a problem? Is C going to work? C will not work. That will not even, if you recover this, you just destroyed your experiment because it's now translating garbage. Okay, so this is a garbage product and this is a garbage product. So this strategy of picking one restriction enzyme is problematic because it already reduces our efficiency of cloning by producing these two other products that we don't want. So how can we make the cloning strategy more efficient? Yeah, you do a double digest. So instead of doing one restriction enzyme, what if we put a second restriction enzyme, a different one, number two. Now you see that all these other products are prevented. So the plasmid cannot self-seal because the restriction enzymes are different. So B goes away. C goes away because the insert can only go in one direction. Okay. So this is a far more efficient cloning strategy, and this is very important. So this is how you clone. You're going to have to pick two different restriction enzymes. Okay. So we want two, and now we need to decide which ones to pick. And the ones that we need to pick cannot cut the gene in half. Okay. So how will we check for restriction sites? Somebody verbally tell me what I should do to check for restriction sites. Actually, let's just pick some quick. Let's pick JO1 because I remember that one. JO1, CT, CGAG, I think. And then what's another one? Hindi3. Let's just Google quick. Hindi3 restriction cut site. Here's Hindi3. Hindi3 cuts at AAGCTT. Let me just write that down. AAGCTT. Okay. So these are, in theory, what I'm going to choose. How can I check them? What should I do? Not a trick question. Yeah, that's the easiest way to do it. That's why I use Word. I like to use Word. So you guys are going to be pressured when you start joining labs and you start doing experiments. There will be these like softwares that will map out all your cloning stuff. What I would encourage you to do is to not invest a ton of time right away in those softwares. I would try to map out your clonings first with Word because you'll really actually learn like what's going on. You'll learn a lot better if you can do it in Word, and then you can maybe like upgrade to those softwares. Okay. So let's just quick control find. So let's search for CTCGAG. That's JO1. It's not in there. Okay. So that's good. So JO1 is check. That one is good. Let's try AAGCTT. AAGCTT. This one's also good. It's not in there. Okay. So we've checked our restriction enzyme sites for internal sites, and we're good. What I did early on is I removed those carriage returns from that initial file that I downloaded. That's an important step because if there is a carriage return, if you had like right here, I'm going to insert CTCGAG. And if you had a carriage return in between that, CTCGAG, it will not detect that sequence. So you have to be careful with your sort of like your syntax, what you're working with. Whereas if you have it in there, now it can detect it. So you just have to be careful. That's why I remove those carriage returns. All right, now it's back to the wild type sequence. Okay, so we know, let's write out our strategy. 5' is going to be Zhou 1. 3' is going to be Hindi 3. Quick just check on the map that that's going in the right direction. Here's the promoter. So here's the directionality is like this, okay? Zhou 1 is on the 5', Hindi 3 is on the 3'. One quick note is when you're picking restriction enzyme sites, never pick two sites that are right next to each other because oftentimes the sites overlap and you won't get efficient cleavage if they're too close together. So it's a best heuristic to choose sites that have good distance between them. So here we're choosing the furthermost one on the left, furthermost one on the right. So that should work. All right. Now we need to design our primers. Okay. So where should my primers be? Where should my forward primer and my reverse primer be? Like here? This should be kind of easy. If we just look back at our map. Where do the primers need to start? Yeah, right at the beginning of the end. Okay, so this is the difference between cloning a gene and just PCRing for a gene. Okay, if I was just doing a detection PCR, like, oh, check to see if this gene is present or check to see if this bacteria which has this gene is present, you can design primers anyway. Doesn't matter. If you're cloning a gene, it actually has to be in frame. It has to be, the syntax has to be all encoded properly. You have to get the start codon, you got to get the stop codon. So I'm actually restricted of where I can clone. If I wanted to, I could pull more sequence upstream and I could potentially clone in the promoter region if I wanted to. Or I could potentially clone in the terminator region 3' if I wanted to. But many times when you're cloning, popping it into a plasmid, you really just want to have crisp edges right at the start codon and the stop codon, okay? So as I design these primers, I know one is going to be somewhere around here. So I'm kind of restricted in my placement. And I know one is going to be here, okay? So my primer is going to be here and here. The question is, how far should I go and on what base should I stop at, okay? So the longer you go, the higher the annealing temperature is, okay? So a good heuristic for setting primers for any PCR is that your annealing temperature should be somewhere between 56 and 60 degrees Celsius. That is like normal temperature that's going to work, okay? So I'm just going to extend out until I hit a temperature within that range. The other rule that you want to follow is what's called the CG clamp rule. Okay, so I've kind of set the argument for why this rule is important in the previous lectures. So when you are amplifying a sequence, here's your DNA. Let's say here's your gene X in the chromosome. When you're amplifying a sequence, okay, your DNA polymerase in the PCR reaction comes on. If you were to zoom in on this, zoom in on this region. Let's say the primer is this long. Let's say here's your gene. Okay. The DNA polymerase, whether it's Taq or fusion, it's probably fusion, is going to come on here and it's going to start writing from this end. Okay. So you really want this end, the 3' end, to be tight, very tight binding. You also want the other end to be very tight binding, but it's not as important as the 3' end because that's where the polymerase actually starts writing. So this is what's called the CG clamp rule, is when you terminate the ends of your primers, you want to end with a C or a G because they have 3 hydrogen bonds, which are tighter than A's or T's. If you end on a bunch of T's or a bunch of A's, you run the risk of creating a primer that kind of is sloppy, like this, where it kind of like the edge hangs off a little bit. It's not as tight. That will not give you as efficient PCR. Okay. So you want to follow the CG clamp rule. Okay. So I'm just going to take a guess. Let's go to the C and let's see what kind of annealing temperature we get. So I'm just going to copy the sequence. On the internet, there is a program, open source, called OligoCalc. I just Google OligoCalc and you'll find this. You can paste your sequence in here and you click calculate and it will give you what's called the salt adjusted annealing temperature. This is the one you want to look at. So it's 58.4, which is pretty good. Okay. So this primer is looking pretty good. The other thing you want to click on is this, check self-complementarity. This is checking for primer dimers and hairpins. I already showed what those were in a previous lecture. And this has none, none, none. So this is essentially like a perfect primer, okay? So we have designed a beautiful primer. Let's mark that off. And let's take this sequence, copy it. I'm gonna start annotating these primers now. So I'm gonna give this primer, this is the forward primer. Let's give it a name. Let's say JFB001 forward. Primer's not done yet, but this is the starting point of the primer. So note here is the forward primer is the 5 to 3' sequence exactly. So when I pulled the gene, I pulled 5' to 3'. The reason you know it's 5 to 3 is because here's the start codon, here's the stop codon. If it wasn't 5 to 3, you would not see an ATG here. You'd see some reverse stop codon, okay? So what you need to remember is that the forward primer is exactly the 5 to 3' sense sequence. It's as simple as that. The reverse primer is where it starts to get a little bit weird. Okay, so let's now think about our reverse primer. Need to figure out how far I need to go in to get something that's roughly around 58. This is 58, what is it, 58.4? 58.4 degrees Celsius. So I'm kind of like noting the features of these primers. Let's try this for my reverse. See what this is at. Calculate 56.6. So I could go further. There's no hairpins, no primer dimers or self-complementarity, so that's good. Let me just quick look at the sequence. If you want to raise the temperature, you just add more bases. So it was 56. That's a little bit lower than this temperature. So I'm just going to see what it looks like when I take 3 extra bases and end with that G. Let's see what the temperature is at. That's 60. Okay. And it's still got no hairpins. I'm going to stick with the hotter temperature. Why would you kind of want hotter primers? So I had a choice. I could have cut the primer off at that G or extend it further and raise the annealing temperature to about 60. One was at 56. This one's at 60. So they're 2 degrees less or more than my other primer. Why would I prefer to choose the hotter one if I can? Are hotter PCR reactions cleaner or dirtier? Maybe that doesn't make sense. Is the annealing more stringent under hot conditions or low cool conditions? Yeah, it has higher specificity when the temperature is hot, okay? So it's going to have higher specificity if you choose the hotter end of the primer as opposed to the lower. The lower you lower your annealing temperature, the lower you lower your annealing temperature, the more sloppy the PCR gets. They can bind other non-specific products. Okay. So I'm going to go with this one, this length, this full length. Now here's where things get kind of tricky. So I'm going to write out the rule. Okay. The reverse primer is the reverse complement of the 5 to 3 sense sequence. Okay. So this is not actually my primer. I have to take this, copy, go to your Google. There's another program called reverse complement. This is just an algorithm that will give me the reverse complement. So I just paste in my sequence, make sure this is clicked to reverse complement, not reverse or complement, but both reverse complement, hit submit. There's my reverse complement sequence. That is my primer. Okay, so let's name this one JFB002. This is the reverse primer. Its annealing temperature was 60, what was it? 60.8. .8 degrees Celsius. Okay. So I got a forward and I got a reverse. The primers have been designed, but they're not complete yet. So we have not yet added in the restriction sites. So in this case, remember that picture, remember this picture that I drew, which is our primers have to look like this. We have to add in our restriction site, and then we have to add in 4 random base pairs. Okay. So let's add our restriction site. Should it be on the left or the right of the forward primer? So that's this one. Should it be on the left or the right? Should be on the left. Yes. Okay. Because that will be, it'll be right here. So we'll be cutting to the left of the start codon. We don't want to cut on the right of the start codon because then we'll lose the start codon. Okay. So let's add our JO1 CTCGAG. We'll give it a little identifier. Now I need to add 4 random bases to allow the restriction enzyme to bind. There's no real rule for this except looking at this primer. Like let's think about this primer. I can pick anything. What would be the problem if I picked this? What's the problem of me picking 4 T's in this case with this specific primer? Does anybody see it? What's the problem? Yeah, I just caused a hairpin. This can now bend over and bind this and create a hairpin. Okay. So there isn't like a set rule of what to pick for these, but you don't want to destroy your primer by adding in a hairpin. So let's not do TTTT. In fact, let's try A. And then we can just take this sequence and we can just recheck it. So let's take this sequence, copy. Let's go back to our oligocalc, paste, calculate, check for self-complementarity. So there is actually a hairpin that's forming, but it's not forming with the 4 random bases. It's forming with the restriction enzyme. Okay. So in this case, let's note there is a hairpin in this primer. What could I do to get rid of that hairpin? If I really was concerned about that, what could I do? You could use a restriction enzyme. So when I started off cloning, I was very paranoid about this. And I would always, if I could find a hairpin, I would go back and I would try to pick a different restriction enzyme. And I would test a whole bunch of different primers until I could design like the perfect primer. I no longer do that anymore. Like I'm too good at PCR. I can just do the hot start. And usually the hot start for me will like fix any problems with any hairpins. So I'm not really worried about the hairpin, but if you were and you wanted a super high efficiency PCR, you could go back and you could pick a different PCR, but you'd have to restart the entire process, check the enzyme, make sure it works, etc. Okay. So I'm going to stick with this. This should be fine, but I'm going to note that there's a hairpin because that's going to require that I do a hot start when I run this PCR reaction. Okay. Now let's try the second one. So now this primer is essentially like, that's ready to go. That's ready to order. $5, they'll ship you that in like 3 days. Okay, let's try a reverse one. So this one's a little bit more complicated. If we were looking at this, just this sequence, which is 5 to 3, which side do we want the restriction enzyme site on? The left or the right? If we want the restriction enzyme site on the left of this or the right? You want it on the right because if it's on the left, it's going to be cutting off the stop codon. So we need to keep the stop codon there preserved. Okay. So the restriction enzyme needs to be on the right. Okay. But this primer is actually the reversed complement. So it's flipped. So in this case, you can just type the restriction enzyme site on the left and you'll be okay. So you kind of treat it like a forward primer once you have taken the reverse complement. Okay. So the HinD3 site was AAGCTT. So I'm just going to type that, AAGCTT. I'm going to make it easier for me to see that. And then now I need to pick 4 random bases. Let's do AAAA again. I would avoid TTTT. Well, AAAA might cause a problem there. So let's try ATAT. Let's see what happens with this. Where is it? Calculate. So this one does have a hairpin, but it's not from my random bases. It's from the HinD3 restriction site. And I don't have a lot of leeway there because I have to specifically clone at this spot. Okay. So I'm just gonna, on this one looks okay, but I'm gonna again note there is a hairpin. So do a hot start. Okay. If I was designing like non-cloning primers, but just PCR primers, I would find perfect sequences within the sequence that have no hairpins. Because you can pick any sequence if you're just doing simple PCR. But in this case, we're trying to actually clone the gene, so we're very restricted in a sense of where these primers actually have to bind. Okay. So we've got these primers. Let me just organize them. So these primers would work. They would clone the gene. How big would the amplicon be? So let's also just talk about primer notation. Once you actually start getting into real molecular biology, you'll design a ton of primers. You'll be designing primers all the time if you get really good at this. So you have to be very careful about your notes, and you need to make sure that you note all the information about these things. So I got the 5' notation. Let's just write like a quick blurb about what these are. These are PCR cloning primers designed to clone Rec-A from E. coli genomic DNA into PBAD-A with 5' Jo1 and 3' HinD3 restriction enzyme sites. That's pretty good notation. I usually also note the amplicon size because that's going to tell you whether your PCR worked or not when you're looking at the agarose gel. How do I find the amplicon size? Yeah, you just count them. So here's the start of it. Here's the end of it. Let's do review if I can get to it. Review word count, 1,062 base pairs. Okay, so I would also just note that the amplicon size is 1,062 base pairs large. Yeah, so we've got notations on our primers. Our primers have names. We got the forward, we got the reverse. That's it. Now let's go over what would happen next. Okay, so we've designed our PCR primers. Next steps. 1, so that's 1, let's do 2. You would run the PCR. You would then, I would quick do like a check. So if we're checking whether this worked, we'd have our ladder. You'd probably be running the 100 base pair ladder, maybe, or maybe the 1 KB ladder. But if you ran the 1 KB, it would look like this. This bottom one would probably be 500 base pairs, and then this ladder would probably be 1,000. Okay, and then it'd go up. This would be 2,000. So your product, if it worked, should be, you should see a band like that. If it worked. If it did not work, you'd go back to that last lecture and you'd kind of go through all those PCR like troubleshooting options until you finally got a band. Okay. Once it was working, then you could take that band. Okay. So you would, once you knew it was working, you'd run kind of like a large PCR, which means like the volume of this thing would probably be like 100 microliters, or you could go up to like 250 microliters if you wanted to do a lot. That's kind of like a lot. You're essentially like purifying a large quantity of this gene. Okay. And then step 4, you would run a PCR cleanup kit. So there's a kit that comes in a box that's has some name like PCR cleanup kit. Depending on the brand, it's going to say something like that. What you do is you take your sample after it's out of the thermocycler. All the Rec-A DNA is in that sample. You run it through this kit. The kit has like a little column that sits inside of a tube. The column has this little white filter. The white filter holds the DNA. So your Rec-A Gene X is stuck to that white filter, and you essentially are able to wash the, clean it up. What are you actually cleaning away? So what gets removed in the PCR cleanup kit? So far, we have not done anything with plasma, not touched any plasmid yet. We've just set up a PCR reaction. So what was in that PCR reaction? Yeah, remember, so there's buffer, there's water. What else is in that? There's a polymerase, probably a high fidelity one. There's DNTPs. There is template DNA, which you definitely want to get rid of, where the gene actually came from. And then there is the, what's missing? Buffer, water, polymerase, DNTPs, template, primers. Okay. And actually now after the PCR, there's your amplicon. So the problem is you need to get rid of all these things. You need to get rid of the extra water, the buffers, the extra polymerase, the extra DNTPs, the extra template, and now your primers. And you just want the amplicon. Okay. So this cleanup kit will do that for you, and then it will produce an elution. An elution is when you drip things off the column. So when you get your elution, now it will just be your purified Rec-A amplicon. Yeah, you can, but if you're cloning, you don't want to do that because there's a loss of yield if you run a gel. Does that make sense? So this is the highest yield way to do it. Although you will have to purify from a gel in a second. Okay, so let's keep finishing the procedure. So now we have, what step are we at? Step 5. So now we have a tube with our purified Rec-A gene inside of it. And where does it need to go? Now's your big chance. Where does the gene need to go? Into the plasmid. Yeah. So in parallel, we had set up some maybe step 0, like in parallel, we had set up a mini prep. We had cultured some bacteria with our backbone plasmid, which is PBAT-A. Okay. We had grown the bacteria. We had done the other kit, which is the mini prep kit. And remember that the mini prep kit, its purpose is to produce a tube that is filled with plasmid. Okay. So now we have our plasmid and our amplicons in separate tubes, and we need to somehow get them together. Okay, so what's the next step to inserting the gene into the plasmid? What needs to happen? Yes, good, exactly. So remember that this Rec-A has restriction enzyme 1 here, restriction enzyme 2 here, and then it's also got some random base pairs and some random base pairs. So the actual amplicon looks like this, and this plasmid is a sealed plasmid. It's uncut, so nothing can go into that if it's sealed. Okay, so the next step, step 6, is a restriction enzyme digest. Okay, so you're gonna set up 2 tubes in parallel. You'll have digestion of your template. You'll have digestion of your backbone plasmid. And what goes into this is essentially a buffer that the enzymes like. The 2 restriction enzymes, typically 1 and 2. Your thing that you're cutting, so your DNA, whether it's the amplicon or whether it's the plasmid, depends on the tube, and then extra water. It's a very simple reaction. You mix these things together, and now you have your cut amplicon and your cut plasmid, okay? Now you have the conundrum of, in these tubes, you've got your cut amplicon and your cut plasmid, but now you've got all this crap. So how do you get rid of this stuff? You have to do another purification procedure. So now what you're learning is molecular biology is a lot of, okay, you mix up a reaction, it does something, and then you need to take a cleaning step to clean away all the other crap to get it to the next stage, okay? So then you run a gel, an agarose gel, and the gel is going to confirm the digest, okay? So you're going to have your ladder, you're going to have your amplicon, and you're going to have your plasmid, okay? Remember, our amplicon is about 1,000 base pairs. Backbone plasmid for PBAD is about 4.5. So this is 2, 3, 4. So this is 5 kb, and this is 4 kb. For the amplicon, you should see this if it all successfully worked. What should you see for the plasmid? How many bands? 2? Why 2? You haven't mixed them together yet. Amplicon is in this lane. So what should you see in the plasmid lane? How many bands? How many bands? Look at it. So look what's happened. This is the plasmid. You digested with 2 enzymes. How many bands? Kind of a trick question. How many bands do you normally see if you run a normal plasmid that's not cut? You see 3. How many bands do you see if you run a plasmid that is cut? 1, because you've cut open the supercoiled and you've cut open the concatenated, so you should only see 1. In this case, in theory, you should see 2, because in theory, there's this thing in between 1 and 2, but in reality, this thing is tiny. It's only like 10 base pairs, maybe it's like 25 base pairs. So you're actually not going to see this band, you're only going to see this backbone. So you should see the linearized, what's called the linearized backbone right here. Just 1 band, because there's no supercoiled, there's no concatenated. So you should see this. If you saw this, then you're like, heck yeah, I'm doing it right. It's going to work, okay? Then what you do is step 8, cut out, okay? So you take a razor blade. That's a razor blade. You'd cut out this. You'd cut out this, okay? You run them through another cleanup, okay? So there's another kit called the gel extraction kit, okay? So it's a little box. Similar thing, it's a column that fits in a tube, okay? And all you're doing is you're taking these cut bands, you melt them, very simple, you just melt it, and then you run it through the kit purification. And what the kit purification is getting away is it's removing the agarose. So now the contaminant is agarose, and you're removing the agarose, okay? And you do 2 of these. So the product of this is that you would end up with 2 separate reactions. One is the cut purified amplicon, and the second is the cut purified backbone. And now we're on to our final step, which is what? Maybe not definitively the final step. Hang with me here 5 more minutes till I finish this. We're very close. If you participate, we can finish quick. What needs to happen with these two? They got to go together. Okay, so now we mix them into one tube, and this step is called now ligation. What does ligase do? Remember back to that first lecture. Yes, it glues the phosphate backbone, or you say it seals the phosphate backbone. So you mix these two together. The ratios depend on what you want to do. My heuristic is usually I put as much amplicon as you can possibly put. So I put a ton of amplicon and a teeny tiny amount of backbone. But in GIST, you mix them together. So you've got a cut amp insert and you've got a cut plasmid. And they're coming together at those two restriction sites, but they're not sealed yet because the ligase has not been added. But you're mixing them together, okay? And then you usually add a buffer, T4 ligase, which is an enzyme from a virus that can do ligation, and water. And then you let this sit overnight at 16 degrees Celsius, which is the temperature that this T4 ligase likes and can ligate at, okay? And then what happens is the ligase will come and it will seal this, and it will seal this, and now it's in there. Now it's a solid chromosome, or not chromosome, but a solid plasmid with your sealed insert, if it worked correctly, okay? Final steps, you have to recover this, okay? So then the final step is you transform the plasmid into E. coli. So you have an E. coli, you grow some E. coli, you do a transformation, which is just a series of steps where in the tube you give them this plasmid, and then they take up this plasmid, and then how do you select for it? You select with resistance markers on the plasmid. So in this case, pVat-A is amp, so then you can do selection. That says selection with ampicillin. Now you have recovered a bacteria that has your plasmid. You can do what's called a glycerol stock, which you guys have already made. You make a glycerol stock, you put it in the freezer, it lasts forever. And then the final thing that you would do is you would do confirmations that you actually got it. So you'd sequence the plasmid with Sanger sequencing, you do a restriction enzyme digest to test whether or not you inserted your gene, and you'd probably just do a gel agarose shift assay to see if it has an insert. So you could confirm in 3 different ways, and then you would be done. Okay, that's it. That's how you clone a gene. That's as simple as that. Are there any questions about that procedure? All right, good work.